Good afternoon and welcome to our webinar this afternoon. Before we start, I'd like to just go through some housekeeping things. So if you have any technical issues, if you can send us a short message in the questions box, which you should see on the right hand side of your screen, we will try to answer them and try to get you back online. During the webinar, RSSL would like to invite you to ask questions, also using the questions box on the right hand side. And at the end of the webinar, time permitting, we'll try to answer as many of these as possible. Any that we don't get round to, we will answer offline and share them with you along with a copy of the slides. So today's webinar is about designing a cleaning validation strategy. There we go. Sorry about that. Technical issues today. So my name's Annette Russell and I'm the Sterile and Non-Sterile Microbiology Lead at RSSL. I've worked in the commercial team here at RSSL for over 6 years, and my role is to support our clients with a wide variety of microbiological testing, all the way through from routine microbial limit testing to the more complex environmental monitoring assessments, or in line with today's topic, cleaning validations, both microbial and chemical. For those of you that are new to RSSL and these webinars, who are we? So RSSL have been around for over 30 years, in fact, almost 40 years, and we are based exclusively in the Reading area. For those of you who don't know where Reading is, it's a town about 40 miles west of London and Heathrow. We have over 250 scientists and supporting clients in 12 different technical areas, of which microbiology is just one of them. And we support clients globally. We even have clients as far away as Singapore and Australia. Our strengths being that we are a relatively compact CRO, so our communication across sites and our site agility to manage complex multidisciplinary projects is superb. We are backed by a multi-billion-dollar parent company, Mondelez, which means we have access to funding to support expansion and growth as well. Our diversity means we're able to support a broad range of biological, microbiological, chemical, and physical analytical services across all phases of clinical development through to commercial release. And we are exceedingly well placed to support all of the new Annex 1 regulations. We are lucky to have a knowledgeable technical team who can advise and support you throughout the process. But enough of me. You're here today to hear Dr. Tim Sandle speak, and I'm sure many of you have heard of Tim and subscribed to his many publications. Tim has over 25 years' experience of microbiological research and biopharmaceutical processing. He is a member of several editorial boards, and he has written over 600 book chapters, peer-reviewed papers, and technical articles relating to microbiology. Tim works for a pharmaceutical manufacturer in the UK and also finds time to be a visiting tutor at both the University of Manchester and UCL. Over to you, Tim.
Okay, thank you very much. Okay, can you see my screen okay?
Yes, Tim.
Brilliant. Okay, well, it's a great pleasure to be back with you all today. And as Annette said, today's topic is all about cleaning validation. And being part of the Annex 1 series, we'll veer a bit towards the microbiological as much as we will the chemical. So the presentation is going to look at cleaning in general. What do we mean by cleaning? Then have a look at some of the approaches to cleaning validation and some of the associated good manufacturing practices. Then we'll address some of the chemical and microbiological concerns that exist in relation to cleaning and validating that cleaning. We'll look at risk assessments, tests and acceptance criteria, and do a bit of troubleshooting about when failures happen in relation to cleaning validation. Excuse me. Okay, so to begin with, what do we mean by cleaning? Well, cleaning is a combination of both physical and chemical means to achieve a required level of cleanliness. So the physical methods include mechanical or manual operations supported by physical techniques such as emulsification and dissolution. Chemical methods include the use of solvents and chemical agents to achieve the solubilization of materials and through the act of rinsing. And cleaning is assessed based on a level of remaining residue that's set below the predefined acceptance level. And these residues can either be found directly on a piece of equipment, or they might be indirectly contained within a final rinse water after that water has been passed over or through a piece of equipment. And in setting the predefined acceptance level, this is to ensure that residues are sufficiently low or to border non-existence, to ensure that the next product manufactured is not compromised by waste or contamination that might exist from the previous product. So we're trying to avoid cross-contamination. And this can be with the residue of the product, or it can also be with any remaining microbial bioburden. So cleaning validation is obviously a key regulatory requirement, and it's designed to ensure that we achieve product and patient safety. And when we're defining a cleaning validation program, we also need to make sure that it is integrated within the quality management system, and that, as Annex 1 requires, that we're using quality risk management principles to help to shape and define the program. So we need to understand and identify risks to patients, which is going to be to the degree of product residue, remaining cleaning chemical residue or microbial bioburden. We need to make sure that we have performed an appropriate impact assessment, that we have a risk mitigation strategy, that we're actually actively mitigating those risks, and we have assurance of finished product safety. And these principles are not only important when we're setting up cleaning validation, we also need to return to them whenever a change is made. So we need to re-review that through the change control process. So with cleaning validation itself, this is a catch-all term for the methodology applied that will give us assurance that a cleaning process has removed residues down to this acceptable level and any other contaminants from a piece of equipment or machinery. So here we're talking about residues from active pharmaceutical ingredients, other process chemicals such as buffers, the cleaning agents themselves. So we may need to also consider detergent. It could be other things that come into contact that are slightly more specialized, like microbial culture media, as we might use if we were conducting aseptic process simulations as required for Annex 1, and also microorganisms themselves. And the assurance relating to cleaning validation refers to both the effectiveness of the cleaning process and also the consistency at which this cleaning can be applied. So can we reproduce what we believe we've achieved on successive occasions? And as with any other type of validation, the validation aspect refers to providing sufficient documented evidence that the acceptance criteria have been met and can be met repeatedly. And in terms of acceptance criteria, as with other forms of validation, we're going to have both critical process parameters and critical quality parameters. So with critical process parameters, the types of ones that are applicable to cleaning validation include time. So here we have the actual process runtime, the time it would typically take to achieve the level of cleanliness that we're seeking, but also we would need to qualify both dirty hold times and clean hold times, and we'll come back to those a little bit later on. We also need to control pressure, particularly during the cleaning process of either the piece of equipment or the fluid that we're using. We need to control the activity, so that's factors like chemical exposure, number of water rinses, and so on. We need to control the chemical, particularly at the required concentration for the required contact time. There's also going to be temperature, because the level of cleanliness we can achieve is going to be proportionate to the temperature at which we're running the cleaning process at. And generally, the lower the temperature, the less effective that becomes. There's also going to be the concentration of cleaning agents. And we also need to have in control things like ensuring that surfaces are still in the validated state. So if we get excessive roughness of surfaces, then we end up with surfaces that are more difficult to clean. And we also need to control flow rate and any mixing requirements. And if we're looking at a manual cleaning process, then we may need additional checks as well, particularly around numbers of rinses and rinse times, and to be able to make periodic assessments of how those undertaking manual cleaning are performing and whether they're practicing everything they should, such as wiping, brushing, and so on. And we'll come back to the concerns about manual cleaning a little bit later. In terms of critical quality parameters, then we're going to have things to consider like remaining product residue, cleaning agent residues, numbers of surviving microorganisms, tests to verify that we've achieved those parameters, and also numbers of rinses and the overall cleaning run time. In terms of when cleaning validation should be performed, obviously it needs to be established for something that is new, be that a piece of equipment or a vessel or something like that in itself, or when it's part of a new manufacturing process. We also need to consider cleaning validation when we're reusing an existing facility or existing process for a different type of product, where we're making major changes to things like raw materials that might be drawn out from an impact assessment, often making significant modifications to cleaning procedures, introducing something new, like a new piece of equipment, into an already established process, making modifications to equipment, if we're making changes to cleaning agents, any change that might impact on the critical process parameter or critical quality attribute of an already approved cleaning validation. And also we may need to make changes in response to regulatory findings. So if a regulator says we haven't necessarily challenged worst case, or that we're not sampling appropriately, then there'd be sufficient to rerun the cleaning validation in order to increase that level of assurance. So just looping back to manual cleaning. Now, cleaning is always preferably undertaken using automated systems because we can better factor in reproducibility and repeatability. But some items, due to their size or bulkiness, or they can't be connected to clean in place technologies, will require manual cleaning, and that's generally through the activity of disassembling and then attempting to clean. So manual cleaning carries a considerably greater risk, and there's some debate you can see in literature about whether manual cleaning can ever really be validated at all, or whether it's more of a case of having sufficient controls to impart a level of assurance. And the reason this is sometimes raised is because there will always be variations between operators or variations with the same operator on different days. Time is difficult to control, and there's going to be variations with the techniques applied, the cleaning materials applied, the quantities of cleaning chemicals used, and so on. So it's often a thing that comes up from regulators that manual cleaning requires a higher degree of scrutiny, a greater degree of checking and other factors to give a level of assurance that the cleaning is up to an acceptable standard. Another variation, another factor that needs considering with any form of cleaning validation is in relation to how many times should a cleaning validation exercise be run. And as with any other form of validation, there's sometimes a recourse to the magic number of 3, which is normally the minimum acceptable level to demonstrate that the results obtained are not due to chance. But to stick only with 3 will assume that a process is consistent and that all the variables are in control. Where that can't be adequately demonstrated, so there may be variances of different types of product where worst case is not necessarily evident, then more than 3 runs would probably be required. But however the number of runs is selected, a rationale that will form part of that cleaning validation master plan will be required. It's also important that the methods used for each stage of a cleaning evaluation must themselves be validated. The other factor to consider is to do with hold times. And here we need to consider time on a couple of levels. So everything in the world of pharmaceuticals comes with an expiry, and cleaning is no exception to this. Therefore, the time frame for the cleaning validity needs to be set, and for dirty conditions becomes critical. So by these, the first thing we need to consider is the equipment's idle time between the end of the last batch and the start of the cleaning process. And this is the dirty hold time. And it's been shown that when equipment is left uncleaned, the longer the duration, the more likely that residues attached to the surface become more rigid. And as they dry over a period of time, they become more challenging for the cleaning process. This also presents greater opportunity for microbial growth as well. So exceeding the dirty hold time outside of what's been validated presents a problem. The second area is the equipment's idle time between the end of the cleaning process and the start of manufacturing. And this is the clean hold time. And this is partly influenced by wherever the equipment is stored as well. So the conditions at which it is, whether it's subject to excessive humidity, and if it's anywhere near dirty equipment and so on. So there's various control factors to consider as well. There are also questions to consider about the appropriate residues to use in cleaning validation. And this can become an issue where equipment is used for the same piece of equipment is used for different products or processes, or for products of varying concentrations. So this might lead to one type of residue being considered to be worst case, or more than one residue would need to be evaluated, or it might be appropriate to use a substitute chemical. Sometimes a substitute chemical that's used is riboflavin, but in these sorts of cases, either the use of a substitute or the selection of worst case requires a justification, and justification will need to consider things like physicochemical binding, viscosity, parts of the equipment that are going to be most exposed, and so on. So a very careful rationale needs to be constructed. And then once that has been achieved, then the methods for assessing the residue should ideally be specific for the target residue. And we'll talk about methods shortly. But if we're going to pick a method that's specific for the residue, then that needs to have undergone appropriate assay validation against assay acceptance criteria, and I've listed those on the slide, but they're the types of things that need to go through proper analytical method assessment. The other area that needs to be considered as well, particularly in relation to the Annex 1 theme, are the microbial aspects. And sometimes this is overlooked. So there's a lot more written about the chemical verification of cleaning. Sometimes what's less clear are the microbial aspects. And important to this aspect are the microorganisms themselves, which can present a direct hazard. We also need to be mindful of the presence of certain residues which are more likely to provide a potential microbial growth source than others, particularly those that could be deemed to be growth promoting for microorganisms. So this also becomes a factor not only for any inherent microorganisms in relation to the use of the equipment, but also microorganisms in the environment where cross-contamination might occur, particularly during the dirty hold period. So certain types of residues might constitute an indirect hazard that we also need to consider and can affect the allowable storage time. The challenge with microbiological assessments is that, unlike a chemical assessment where equipment can be deliberately soiled with the residue of choice after we've done the appropriate rationale, we can't deliberately introduce microorganisms into the cleaning process. That would be deemed to be a poor practice, partly because demonstrating complete removal would always be challenging. So when we're trying to attempt a microbiological assessment, we need to make sure we've got a very good sampling plan to account for this sort of lack of direct evidence. Now, the types of cleaning agents that we wish to select will have different effects upon the target residue that we're trying to remove, and also upon any surviving microorganisms. And this is going to be a factor of the chemical, the residue, and the species and numbers of microorganisms that might be present on a given surface. Now, such cleaning agents tend to be one that's a caustic-based agent, such as sodium hydroxide, and this may or may not be used in combination with an acid-based reagent, such as hydrochloric acid. Now, for the microbial aspect, unlike disinfectant efficacy, these agents are not normally subjected to the same level of robustness that other forms of disinfectants would be subjected to, but it's still important that we undertake a sufficient level of post-activity testing in order to assess their effectiveness. There's also many commercial cleaning agents which are formulated for cleaning with surfactants, and they may well have more like to have an alkaline source and a chelate. Now, with this issue about acids and bases or caustic agents for cleaning validation, in terms of microbiological concerns, then literature strongly suggests that alkaline chemicals are the most effective in removing organic soils, so against target residues like oils, fats, proteins, starches, carbohydrates, and so on. So most of the common types of residues that we would come across within the pharmaceutical sector. And these work by generally hydrolyzing peptide bonds and breaking down large insoluble proteins into smaller and more easily soluble polypeptides. And these types of chemicals are also relatively effective against most types of microorganisms, provided they can make contact with the microorganisms. So we would sometimes need to ensure that we've removed sufficient residue, and that if there are any remaining microorganisms bound to the surface, that the alkaline chemical like sodium hydroxide can come into contact. With the acid chemicals, then whether they are needed or not partly depends upon whether any heavier soils are present, or anything like beta-glucans, which are going to be more resistant to the alkali chemicals, and also whether there were any problematic microorganisms, or there was a need to neutralize the use of the alkali chemical. So acids like hydrochloric acid or phosphoric acid are available for that. In terms of water rinses, that's another key consideration. So water rinses are very important for removing chemicals that have been used for cleaning, because there's little value in using a chemical to attack a product residue and to leave the cleaning chemical behind. Water also has a very key role to play in the microbiological aspect by helping to siphon away those microorganisms that have become disassociated from the surface. And water also helps to keep soil suspended away from the clean surface, so it then starts to function as an redeposition agent. There's also some important characteristics of both water and of the cleaning agent. So cleaning agents that have the ability to wet a surface and penetrate the soil deposit are important. Those that have the capacity to break soil into fine particles, that can hold fine particles in suspension, and then there's sufficient motion, pressure and speed to prevent redeposition. The final water rinse should be of the same chemical or microbiological purity as the source water. So if we're using purified water, that gives one set of acceptance criteria. If we're using water for injections, then it gives another set of acceptance criteria. And if our requirement is to use water for injections, and let's say it's later stage processing of equipment to be used in sterile manufacturing, then we would also need to assess levels of endotoxin as well as microbial bioburden. It's also important with the aspect of water is that when the cleaning process is finished and the final rinsing has been completed, that we do not leave any piece of equipment with residual water remaining. And that's because that provides an opportunity for microbial growth, and particularly any cross-contamination from the environment in which we're holding the piece of equipment. So the last step of any effective cleaning procedure should involve demonstrable drying. And this could be the addition of a solvent, such as 70% sterile isopropyl alcohol, or flushing with sterile compressed air, or some other mechanism. But what we're trying to do is to ensure there is no subsequent opportunity for microbial growth. It's important that all cleaning validation approaches are based on risk assessment. And as I mentioned at the beginning, this is one of the key Annex 1 requirements. So we need to look at a whole series of risk factors and to understand variability and uncertainty and lack of reproducibility that might be present in the process. And the types of worst case factors that we need to consider are going to vary to an extent on the stage of manufacture, the type of soiling that might be present, the level of expected residues. We need to have a thorough understanding of the design of the equipment, particularly the ability of the cleaning chemicals to penetrate to every product contact site, the ease at which equipment can be dried afterwards, the surface materials, and particularly the age of the equipment and the level of damage to any surfaces. So particularly if we've got something constructed of stainless steel, if that is scratched, then every scratch provides a greater opportunity for residues to become deposited or pockets for microorganisms to take refuge and begin to build biofilm communities. So there's lots of factors that need to go into a risk assessment to help build the worst case. So the build-up of worst case comes from the risk assessment and ensures that the cleaning validation process is sufficiently robust. Another factors to consider in this worst case assessment, as particularly for equipment that's used for multi-usage, would be drugs with the lowest solubility against a cleaning agent. We need to consider locations that are difficult to clean, particularly for targeting swabbing, and it's not something you can really turn around to a regulator and say, yeah, we've got a location that's difficult to clean, but we didn't sample it because it's difficult to get to. That then makes it a location that you really then got to try and get to and sample or disassemble in some way. We also need to consider if we've got similar items of equipment, those that cater for the largest number of products, we may wish to consider drugs with higher toxicity as well. So there's other things to feed into the risk assessment process that we're building up with this worst case situation. Also, the risk assessment will need to consider the stage of the process, which I mentioned earlier. But with this, the level of cleaning and the level of acceptable remaining residues can sometimes relate to the stage of manufacture. So with the use at an earlier stage in the process, this may, and I'm emphasising may, require lower levels of cleaning compared with equipment that's used for product at an intermediate or a final stage. And this partly depends on the factors of likely carryover or not, but it's something that needs to be considered. It's often also the case that levels of cleaning need to be greater if the cleaning is to be followed by sanitisation or by sterilisation. And it also stands that the tolerable level of microorganisms or the types of microorganisms will differ also between sterile products and equipment used in non-sterile manufacturing. So there are things that need to come into the risk assessment, and building on that microbial aspect, using the risk terminology of severity and likelihood is also quite useful in this situation. So the microbial hazards should be identified, and we should have an understanding of the likely types of microorganisms, and then consider their relative risk based on the severity and the likelihood that hazards will that can present themselves. So we need to be mindful of microbial contamination on the equipment post-use and pre-cleaning. We need to be mindful of the whole time prior to cleaning and the chance of microbial proliferation. And this also has association with the potential release of endotoxin. Any additional microbial challenges from the storage environment. So that's environment to equipment recontamination. The efficacy of the cleaning process to remove microorganisms and endotoxin. And also then, as I mentioned, the storage of the equipment after it's been cleaned. So all of that also needs to be built into the risk assessment process. And certainly the likelihood of contamination is partly going to be a factor of equipment design and also the easiness of cleaning. Now, I mentioned worst case conditions for testing. And here we need to ensure that we're undertaking the sampling for cleaning validation at the end of the cleaning process, immediately at the end of cleaning, so that we're getting a more direct assessment of the ability to effectively clean using the defined process. It's also important, though, that we're not starting the cleaning validation until we've reached the point of the maximum hold time. And that's because of that point I mentioned earlier about dirty equipment being harder to clean and soil becoming more difficult to remove, and also the greater chance of microbial contamination build up. We also need to consider, just back to the question of residues, is that the residue that we have in mind for chemical cleaning validation, which might be, say, based on the most viscous substance, for example, may not necessarily be the one we'd be most concerned about for supporting microbial growth. So if we're using a vessel for product and for culture media, that might be the situation where we need to run two types of cleaning validation, for example. The locations for monitoring is also another very important consideration, and these need to be representative of the contact sites that the product has with the piece of equipment, and also of the worst case locations in relation to cleaning effectivity. And we need to be aware of things like non-uniform contamination transfer. So we might want to differentiate between equipment where the potential for contamination is relatively uniform. So some vessel that mixes, contamination could get any single one location could be equally representative of another, and differentiate that between where there could be non-uniform contamination. So contamination might be lodged in a filling needle or in a certain part of a compressing machine, for example. And these locations, once selected, need to be defined in a protocol. Each location needs to be justified using risk acceptance criteria. And we should avoid the regulatory charge that we've simply picked locations arbitrarily or gone for the easiest to clean locations. We also need to include both swab and rinse samples, and the data from both may not necessarily correlate, particularly in a situation where a failure occurs, and there are some differences. So the swab will always focus on a small area, the rinse gives information about a larger area. The swab can directly measure worst case if we're picking the appropriate location, so it'll be more representative, but the rinse can give us an average of the remaining residue or bioburden. It can be that if a swab fails, a rinse may pass. That's natural because of the greater dilution from the rinse. But if a rinse fails, it's also quite likely the swab will also fail. So there'll be a location of concern if we've picked the right swab locations. We also need to have appropriate limits. Now for chemical cleaning, there is considerable advice based on different concepts. So one concept is the maximum allowable carryover, which mathematically expresses how much of the previous product is likely to carry over to the next product. Or alternatively, there's the no observed effect level, which informs us that the drug quality that has no observable effect on human health exists when assessed at that point. And there are different criteria that spring from that. So one set of criteria is to have not more than 0.1% of the normal therapeutic dose of any product as appearing. Another is to have not more than 10 parts per million of any product appearing likely to cross-contaminate to another product. If it's very early stage, or if it's through something that's less significant, then you can simply look for lack of visible residues, but that wouldn't be acceptable for product contact materials. And there are different methods to assess, and earlier I mentioned about specific and non-specific methods. So non-specific methods are things like conductivity, pH, and total organic carbon, and each one of those have their limitations. They're good for low-level detection, they're fairly rapid, and they're inexpensive, but specific methods are far better and will tell us a lot more information. So things like titration and chromatography, electrophoresis and ELISA are superior methods for assessing residues. When it comes to microbial limits, again, choices need to be made, and there's going to be differences between sterile and non-sterile processing. So in non-sterile processing, there's likely to be greater emphasis upon objectionable microorganisms. With sterile processing, we want to have low numbers that are not going to present a sufficient challenge to the sterilization process. And often we can use water for injection levels based on final rinses. So we want to see bioburns of less than 10 CFU per 100 ml and endotoxin levels below 0.25 EU per ml. For non-steriles, then there's a very good paper now, sort of getting close to 25 years old from Doherty, and that gives some very good formula for assessing the permitted microbial levels from working backwards from the finished product through the process, and it allows an expression of colony forming units per square centimetre. So the various things I've said, the various factors, all then need to be put together, and this then allows us to establish the cleaning validation process. And I've tried to summarise that in a flow diagram of the various steps that need to be considered, which I won't go through now for the sake of time, but it's something you can have a look at, and hopefully that will present a good checklist for when you're putting together the cleaning validation and the associated SOPs. As with any form of validation, the cleaning validation should be appropriately documented. And on the slide there, again, for you to read it, as a takeaway from the webinar are some of the key criteria that need to be included in the cleaning validation documentation. And in particular, the use of checklist is really important, and this can also help in terms of structuring regulatory submissions. It's also important that the SOPs that come out of the cleaning validation, the actual documents that operators are expected to follow, reflect what really goes on in the cleaning validation, so that they're covering things like how to disassemble equipment, the right cleaning method to be used, the detail of the concentration of the cleaning agents, controlling things like flow rate, pressure, rinse times, and that people are appropriately controlled against this. And one of those key controls, to loop back to something I was saying earlier, is linked to time. So we need to build time into all of the controls. So minimizing time, not exceeding dirty hold times, and segregating clean equipment, and ensuring that's used in good time as well. Then just to finish with, there are some challenges with the cleaning validation process and some things that can trigger failures. So anything to do with manual cleaning really is challenging. The design of CIP systems, particularly if there are dead legs and lack of drainability, can be an issue. Sometimes gradual build up over time if we're not adequately assessing and seeing this happening, particularly with physical protein, presents a problem. If aspects of CIP operation becomes faulty, like blocked steam traps, or we have poor maintenance practices, then that can cause operational challenges. Another concern is around not developing appropriate acceptance criteria, failure to undertake risk assessments, and failure to assess things like air-liquid interfaces, or accounting for surface damage. And this can allow microbial deposits to build up. And one of the biggest concerns here is with rouging. So rouging, there should be checks for rouging within equipment, and this can lead to not only the build-up of particulates, causing chemical reactions through the rouge product that might be unexpected. It also causes corrosion and pitting, which leads to opportunities for microbial colonization. And then just building back to the issue of failures, as well as some of those variables, if we have a failure with the rinse water, this is often easier to address than direct contamination on surfaces, because we can increase the number of rinses, or we can slightly alter the chemical treatment. Where we're getting direct recovery of residues of microorganisms from surfaces, then that becomes more problematic, because we may not be getting sufficient contact of the cleaning solution with the particular risk area of the piece of equipment. And we also need to consider operator variability. Okay, so this brings the webinar to an end, and I know there was an awful lot of material to consider, but hopefully there's some things to consider, particularly if you need to additionally consider the microbial aspect, which fits in with the Annex 1 theme of this webinar series. But there are a lot of variables. The key things are sufficient risk assessment, sufficient acknowledgement of the appropriate residues and hard to reach areas, and building reproducibility into the process. So time spent developing a rationale, and the risk assessment is perhaps the best time spent and the thing that can stand up most greatly to regulatory approval. Okay, so that brings the part of the webinar to the end, and I'll pass you over to Annette.
Thank you, Tim. I'll just so we have time for some questions. Just a couple, because that was quite an intensive webinar. So I have a question from Vinod that said, can you advise on the type of swabbing techniques people should use, and how to take swab samples where you physically can't reach?
So that's always it's always a challenge. So to answer the last bit, there are various like extension devices that you can buy to try and extend the swab, but it isn't easy. But I have had experience of regulatory inspections where we tried to use the argument that we can't necessarily get the swab to a certain location, and they've said, well, no, we believe that to be worst case, so you'll have to either find some way of extending the swab or go through some disassembly process. In terms of swabbing technique, it's always important to be able to rotate the swab head as much as possible. It's important that if the swab is it needs to be moistened, and for microbial sampling, we need to make sure that the swab has some kind of neutralizer or some neutralization effect in case there are traces of cleaning chemicals.
So sticking on the themes of swabs, would you use swabs or contact plates for microbial sampling?
I think it depends on the nature of the surface. In most cases, we need to resort to swabs. The reason some people try the contact plate is generally they have superior recovery. Although if we're using flock swabs, then these tend to have the ability to capture more from a surface. So given most pieces of equipment, it's normally default to a swab, but there are some cases where a contact plate could be considered.
So this is probably the last question we've got time for. It's from Annika, and she says, if you have a product that does not come into direct contact with equipment, is residue testing still necessary?
Yes, but the residue would perhaps be more orientated towards the cleaning chemical, because obviously you still need to clean, and you've got to prove that you've got rid of traces of detergent or alkaline or acid-based chemical, but it might be something different that you're looking for, and that's important to build into the risk assessment before you undertake the cleaning validation.
Okay, well, thank you, Tim. I'm sorry to all those people whose questions we haven't managed to answer. There were quite a few that come through at the last minute. As I said before, I'll pass these on to Tim, and he will answer them offline and send them out with a copy of the slides. Okay, so I hope you've enjoyed the webinar. I hope you found it useful. And as I said, I'll pass on those questions and the slides. Our next webinar is on the 24th of May, where Tim will discuss how water systems can go wrong and what to do about it. As always, spaces are limited, so don't forget to register. And we have some training courses, particularly around the cleaning validation, the environmental monitoring and the water systems in the next few months, which may be of interest. So again, all details can be found on our website, or you can contact me direct. If you have any questions or any queries, or you can't find anything on the website, please feel free to drop a line to me. So that's it for today. Thank you for attending, and we look forward to seeing you on the next webinar in May. Thank you.
Thank you.